before last time we were talking about the tumor environment, and so now we're going to talk about one of the next hallmarks of cancer. And it's important for several different reasons to really get a handle on this. The first, and I guess the most important really, is the fact that tumors can't get, which I mentioned, I know, they can't get big enough to cause problems unless, of course, you have blood vessel supply. Okay? And the blood vessels, as we'll talk about, they're not normal. The blood vessels that tumors do develop are abnormal, and people are taking advantage of that to design treatments. But one of the major problems, and which again we mentioned earlier, but we'll talk about next, kind of, metastasis, is that when the blood vessels feed a tumor, and it's not clear who's invading whom, right? Because is it the tumor invading the body, or are the blood vessels invading the tumor? It's not clear, right? They're probably kind of both happening. But the blood vessels, once a tumor becomes vascularized, it is immediately provided with a highway system that goes everywhere else in the body. Okay, and so it's not just blood vessels, it's also lymphatic vessels, and we'll talk about that. So the first slide, of course, looks like blood vessels and capillaries. It is, of course, a tree, which look the same. And they look the same for a similar reason, right? Blood vessels spread out to cover as much area as possible. In the case of branches, you're harvesting as much light as you can. In the case of blood vessels, you want to be able to distribute oxygen and nutrients as widely as possible, right? So it's not an accident that they look similar to each other. Okay? So angiogenesis, first of all, people tend to think of things that happen in cancer. Frequently, they think of them as being all bizarro and abnormal. But you're learning that what cancer does is take a normal process, be it inflammation, be it division, migration, and it twists it, right? It happens when it shouldn't be happening. And so that's really what's going on. So angiogenesis is a normal process. It occurs during development in response to injury, disease. As I mentioned before, the cyclic fashion during menstruation, right? The tissue in women the uterus is prepared to receive the implanted embryo. If that doesn't happen, that whole thing gets flushed and you make a new one, right? Every month. And that involves, of course, angiogenesis. And cells that are more than just 1 or 2 millimeters away from a blood vessel can't grow. Okay? So this just shows kind of like the distance away from a blood vessel. And then when you start to get further away, those cells start to get sick. Okay? Here's another blood vessel with its ring of nutrient delivery. But the angiogenesis that we see during development, like during the development of a baby or any other organ or tissue, is programmed. And in embryogenesis, it's a programmed sequence of events that happens. In angiogenesis and cancer, it's dependent on local signals, and that's actually pretty important, and that's probably one of the reasons why the blood vessels that develop in in tumors are so abnormal. And the term that you'll see in the literature, if you actually read the cancer literature, and you're going to see the term, they say that the blood vessels are tortuous. And what that means is they're tortured or tortuous, meaning that they are much more curvy and loop around, and instead of that nice branching pattern, right, with a bunch of parallel lines, ultimately a bunch of things reaching out, they loop back all over the place, so they don't grow normally. And one of the things that we'll talk about when we talk about treatments is the fact that the blood vessels are leaky. Okay, so again, So blood vessels are typically very tight. You have a small amount of fluid that, due to the blood pressure caused by the pumping of the heart and contraction of those muscle, the myofibroblasts that I mentioned before, those contractile fibroblasts, they line the outside of the blood vessels and they squeeze it, and that's what gives the pressure inside the vessels, allows it to be pumped around the body. But a small amount of fluid can leak out of a normal blood vessel. With tumor blood vessels, you have much more fluid that's able to cross, right? So you have a net movement out of the blood vessels and into the tissues. And again, that causes some problems that we'll talk about, right? I mean, these are things that are all directly clinically relevant to patients. It seems like trivia to say they're leaky, but it makes a big difference in trying to give someone chemotherapy. Okay? So we'll talk about them some more. So just to see the picture, in a healthy vessel, you get this branching pattern and long runs of a space in between the branches. You get these long runs. You get a branch, it goes off, and the tumor vessels look like this. You don't get even spacing. You don't get nice coverage. They loop back on themselves. They're, you don't need to be a super specialist to look at these two pictures and see that they don't look alike. Okay? But they don't have, they don't have the normal cells lining the outside, right? So there's multiple layers in a blood vessel, which we'll look at. There's the endothelium, the inside. Endo means within. Endothelium, we have those cells that are lining. And then you have cells on the outside that are giving support, structure, contraction, things like that. The tumor blood vessels tend not to have those the way they're supposed to. Okay? So even though tumors do get a blood supply, it's not normal. It doesn't look normal. And so here is another example. These are essentially spheroids, balls. MCF7 is a breast cancer cell line. So these are breast cancers that are growing. And so this is just MCF7. MCF-7 is just a clone of this MCF breast cancer. And you can see that here we have the long, stringy, more normal looking blood vessels. Here, when you have overexpression of HER2, the growth factor receptor GRO2 in this same ball, remember, this is not a real tumor, this is a model. But in this model, if you overexpress a growth factor receptor, you get that it very much mimics that tortuous, folded up, looped around look. Okay? So one of the critical things in angiogenesis is a growth factor called VEGF. Right? That's how people say it, VEGF. And it stands for vascular, it's written out here, vascular endothelial derived growth factor, VEGF. Okay. And VEGF binds to its receptors. There are 3 of them that we know of, VEGF receptor 1, 2, and 3. There are at least 5 different forms of the growth factor itself. Alternative splicing, processing, there's multiple forms of VEGF. It's not yet clear what all of the different forms of VEGF are doing. Right? VEGF-A is the one that people study the most. The receptors for VEGF are all receptor tyrosine kinases. And we talked about those earlier in the course, right, when we talked about oncogenes. So all of these receptors, when they bind to VEGF, are going to become active tyrosine kinases. They're going to phosphorylate target proteins on tyrosine. Okay? And the VEGF, this growth factor, which causes the formation of blood vessels, also causes increased vascular permeability which then, of course, leads to leakiness. Right, so if you have a lot of VEGF around, those blood vessels are going to leak fluid more. And that changes the pressure inside the tumor. Right? If you have more fluid outside than inside, that's going to cause changes. And that actually affects treatment. Okay, so again, here's the VEGF family, not to be memorized, right? Please. Even if I was going to test you, which I'm not, you still wouldn't memorize this. Okay? But suffice it to say, there are multiple forms of VEGF. VEGF A is the one that people study the most because it's the one that binds to the receptor on cells that leads to angiogenesis, vasculogenesis, and vascular permeability. Okay? And these are related terms. Okay? VEGF C and VEGF D seem to be more responsible for the development of lymph vessels, right? So the lymphatic system and the circulatory system are derived from the same precursor cells, right? With the main difference is the fluid that goes in them. One has blood, the other has lymph. Right? But they look like each other and they work very similar to each other, including overlapping growth factors. Okay? So here is vasculature from a wild type mouse. You can see that you get this nice looping. You get very even coverage. You could almost measure the intervascular space or something, right? You could set up a grid. Whereas here you get vasculature from animals that are overexpressing VEGF. You get all kind of more vessels, but they don't have this same nice pattern, right? You get this wiggly stuff going on, right? As opposed to more linear stretches. Can everyone see that? Okay. And VEGF is not the whole story. So there are a bunch of different regulators of angiogenesis, and you watched. The two guys came in later. Did you guys both watch the Cancer Warrior?
Cancer Warrior.
Yes, I did.
You didn't, did you?
No, yes, I did.
No, yeah, you did. He looks guilty. Looks very guilty. Yeah. So if you saw it, you know that there are both activators and inhibitors. There's a bunch of growth factors which are able to stimulate angiogenesis. In one reason that the HER2 overexpressing cells had that weird angiogenesis is that is the growth factor here. The epidermal growth factor is what binds to that receptor. Okay, so there are things that turn angiogenesis on and a bunch of things that turn it off. Bottom line, it's a complicated system, right? With a bunch of things that are turning it on, and also multiple things that are turning angiogenesis off. And so here, I wanted to show you because I think this is not only the present, but the main future. If you guys go into the clinic and stuff, you're going to be dealing with this kind of thing. The MERS, the microRNAs. Okay, the microRNAs. Because this is something people didn't know about until the last maybe 10 years, really understand what's going on with these non-coding genes, right? That are making these microRNAs. And so this is just an example for you. I just pulled a quick thing, right? So that you can use this anti-MER21. It's an antagomer, so it's a MER that blocks another one. As a therapeutic tool, it perturbs angiogenesis. This one, microRNA 542, whatever, inhibits angiogenesis, and this one responds to hypoxia. So the microRNAs play a role as well. Right? The established players are the growth factor proteins and their receptors, but we now know that inside the cell, the regulation is really important. Okay, and here is another one, right? Upregulation of this particular microRNA promotes tumor angiogenesis by targeting the von Hippel-Lindau tumor suppressor, and it's associated with poor prognosis and triple-negative breast cancer. So these microRNAs are going to turn out to be key, right? When you guys are treating patients, when you guys are docs, it's going to be all molecular, right? There's not going to be throw this person chemo, everyone treated the same. It's already changing, but the future is on the wall, right? The writing is very clear that this is what's going to happen. And so these microRNAs, I think, are going to enter the picture, right, as to something that's taken into account when you're trying to give someone a diagnosis and decide on a treatment course, okay? And an antagomere, this is just bio slang for you to learn, right? An antagomere is something that inhibits the activity of a microRNA. Antago, of course, is antagonize, or to block. So an antagomere. Okay. And angiogenesis, of course, as we have talked about, has a direct link to inflammation, right? So this VEGF receptor signaling is a link between inflammation and tumorigenesis. So inflammation drives the activity of some of these cytokines, including the growth factor VEGF, and VEGF receptor signaling is linked to inflammation. Okay, so we have crosstalk between angiogenesis and inflammation. And inflammation, we know, is critical. Right.
Okay. Is that like the signaling process somehow causes inflammation, or how does it work?
Yeah, I mean, I think inflammation more likely leads to the production of VEGF, and VEGF leads to the activation of its receptor, and they talk back and forth. Okay, so, and then, of course, there are behavioral choices that influence things. So here we have, everybody recognize this guy?
Is he from Panama?
Yes, this is Don Draper. Right? So, of course, in the 60s, everyone smoked. My mom smoked when she was pregnant with me, right? Back when that show was filmed in the U.S., everybody smoked. It was very suave and sophisticated to smoke, right? Okay. And here is an example. This is actually an older article. Secondhand smoke stimulates tumor angiogenesis and growth. So the environment can actually influence even the development of blood vessels in a tumor. Okay, and why exactly that is, I don't know, but you have an increase in the size, you have increases in VEGF, right? And so it's they're linked, right? Now, a thing that's really important about all this, like I said, tumors can only get to be a teeny little size unless they have a blood vessel. So angiogenesis directly controls the size to which a tumor can grow. And a tumor can stay in a dormant state for really long periods of time, right? So tumors can stay in what's called a dormant state. And then what seems to happen is in any successful tumor, there may be tumors that never make the change. We just don't see them, right? They're too small to see. But for those tumors that grow big enough to cause a problem, the term that's used is it's the angiogenic switch, right? That is a technical term. Sometimes I make up crap, but this is a real one. Okay? So the angiogenic switch is the term, and that's essentially an on-off kind of thing. It's essentially a binary thing. This thing is either able to stimulate blood vessels development or it's not. And if you have increased angiogenesis, it correlates with a worse prognosis. Those of you who watched the Cancer Warrior, right? You saw Folkman, right? He was a surgeon, and he said that people were saying angiogenesis wasn't important in tumor development, but if you remember, I'm paraphrasing, but he says, I handled tumors and I knew they were bloody and hot, right? That these things had a tremendous vasculature, right? And the answer is you cannot become a life-threatening tumor unless you have a blood supply. They just can't grow. Yeah.
Does that have any relationship with when they say that a tumor is benign? I don't know how you say it.
Benign?
That
A benign? No. You can have. That's a great question. So the difference between metastatic and benign is not dependent on its size. You can actually have a massive benign tumor, so that for some reason these things always happen in Eastern Europe. I guess these women just walk around with these massive things hanging off their bodies or something. Hold on, Yalda is calling in, so let me... This angiogenic switch that we see, this ability of tumors to develop a new blood supply, is definitely correlated with a worse prognosis and the size of the tumor. And it seems to be based on a balance of things. We have VEGF and we have this and we have this, but then if you watched the video, you would say, oh, but we have endostatin and we have angiostatin, right? We have these things that block angiogenesis, and there's a balance in there in which you have essentially activators and inhibitors that are canceling each other out. But at some point, the tumors will either produce their own factors that are allowing them to grow, or they will trick nearby cells. Very often it's the stromal cells, the cells in the nearby environment, that are producing the growth factors and that are allowing the tumors to grow. So remember, we said that the stromal cells become abnormal, right? And one of the things that they do when they start to act abnormally, right, is to begin to produce angiogenic growth factors. And so you have a story here, right, in which you start here on the far left with a happy purple cell. Yeah, purple my favorite color, people. So yeah, I'm wearing purple. So you have your happy purple cell, and then you have some genetic alterations. Those genetic alterations can lead to the death of the cell. Or you can have the formation, right, of this small, less than 2 or 3 millimeters. So again, a very small dormant tumor. And these dormant tumors seem to be not only a problem in the development of a primary cancer for people, but they seem to be a major problem for people in recurrence, because it's very hard to treat a dormant tumor. A tumor that isn't growing very much doesn't respond to treatment the same as one that's growing very quickly. And so if you have these little clumps of cells that are scattered around the body, they're called micrometastases. Right? Micro meaning very small, metastases. And we talked last time about the fact that tumor cells often will leave the initial tumor in clumps. And so you get a small clump of cells, maybe you get some stromal cells, some cancer cells, you form a small dormant tumor somewhere. This thing doesn't get a blood supply, and it's very difficult to reach and kill with the cancer treatments. Okay? And so you have, one way or the other, a switch gets thrown in a small number of cells and the original formation of a tumor, or after you have a micro tumor. Either way, the only way you're going to get a tumor that's big enough to cause problems for a person is if you get this blood supply in there to those tumors. Right? It doesn't happen any other way. right? It used to be thought, by the way, that cancers like lymphoma, they used to think, oh, that's a blood cancer. It would not need a blood supply. And we now know that things like multiple myeloma, lymphomas, that anything that forms a mass, even if it's in like a lymph node or spleen, they actually do depend to an extent on development of new blood supplies, even blood cells, right? So they still use them. Okay, so you asked, and this is where I was starting to talk, so I'm going to show you a picture. Actually, right when Yalda called in, we were talking about benign tumors. Sorry, actually, I forgot, but this is a good example. Here is a woman who had, here's her tumor.
Wow.
Okay, it looks weird like that because they've, covered her with iodine and everything to make it, to try to sterilize it. I mean, it wasn't that color before, right? But this tumor, right, which is enormous, right? How big did it say it was? 303 pounds. Okay? So this person had a 303-pound tumor. I think the rest of the person, I bet she only weighed 100 and some pounds, right? So this is an example, an extreme example, obviously, of a benign tumor, because you would never have a malignant tumor that got that big without killing you, right?
How is it possible that somebody waits for so long until the tumor is 300 pounds?
Yeah, I mean, I know she was bedridden for a while. If you read the previous slide, it'll give a little bit of her history. This wasn't occurring in a place where they have good medicine, obviously, and she was bedridden with this thing. I mean, so should she have done that? No, that was probably a bad decision. She should have gone to seek help, but she may not have felt like any was available, and she might have been embarrassed, and then it got really big. I don't know. Right? But you do see these things. There was just a paper in the news a couple days ago. There was a picture of a young African girl, a 16-year-old girl, I think, and she had a big tumor like that with her teeth were in it and everything, like it was coming out of her jaw. But she was somehow, I don't know how, but she was still able to eat. But see, this is actually the tumor they took off this woman. Look, it took how many people to move it off?
Wow.
Okay. So this tumor had a lot of blood vessels, right? And that's one of the, obviously, when you have a tumor this big, it's one of the problems with getting it off is to do it without letting the person bleed, right, to death. Now, benign tumor means that this thing was obviously huge. So benign isn't related to size, right? But the difference between a benign tumor like this one and a malignant tumor, which can kill you much, much smaller, right, can kill you, is the fact that malignant tumors, by definition, invade nearby tissues. If you look at this, what does this tumor look like? One of the big hallmarks, this is a big round blob. It looks like someone took a balloon and blew it up, right? And it does, of course, has some indents and folds. It's got some places where it dents in. But doesn't this look like a big old sack, right? And the reason that happens is that the membrane that surrounded that tumor when it first started to form in the ovary, it never burst and punched its way out and spread, right, into those other things. That involves those matrix metalloproteases we talked about before and we'll talk about again. But a benign tumor stays in its original location. In this particular case, the location was in this woman's abdomen. And so there's room for it to grow, and then it slowly pushed and forced its way, right? So that it was, she had a great big belly, right? You're not going to get a big benign tumor in your brain because there's no room. Does everyone get that? Mm-hmm. So you tend to see these large benign tumors hanging on the outside of people, as like hanging on their skin or in their abdomen or something like that. Right, where there's room. Okay. So now I was saying that the tumors, the blood vessels that form in the tumors are abnormal. And tumor angiogenesis, because it's abnormal, leads to some differences that affect the response to treatment, right? I mean, we never can forget why we care about this disease, right? It's people. It's the ribbon that you held up, right? That's why we care, right? Is the people. And this angiogenesis absolutely affects things. So the first is that because the tumors are leaky, more fluid gets pushed out into the space around there than in normal tissues. And the lymph system, the lymphatic vessels, their normal function in our body is to retrieve the fluid that leaks out of the blood supply, and they collect that. It drains into lymph nodes, it goes into bigger and bigger vessels, and it gets dumped back into the circulatory system up in your shoulder, right? In your right up here, right? There is a big lymphatic vessel that dumps back into the circulation and returns that fluid, right? That's why you don't swell, your body doesn't swell up. But inside the tumors, the lymphatic vessels are abnormal, and so they have a high pressure. The term for that is interstitial, between the tissues, between the tissues, you get this interstitial pressure. And so there's multiple reasons. One, the vessels are leaky, which we talked about. Two is the cancer cells are actually crowding out the vessels and squishing them, right? So they're increasing the pressure in that area, and the fluid leaks out. You have poor lymphatic drainage, which I mentioned, and then you have increased amounts of the platelet-derived growth factor, which can cause cells to just squirt, release fluid. So there's multiple things that are pushing fluid into that. The pressure difference between the blood and the tissue then is decreased. Does that make sense? So normally we have more blood pressure in our blood vessels. That's one of the reasons why the oxygen and things gets forced out of the capillaries, right? You have the exchange. But because of the increased pressure in the tumors, the difference is decreased. And when you don't have that same amount of pressure, you have less movement of fluid out of the blood vessels at that point, right? There's a new equilibrium. Are you with me?
I lost.
Right? So, because I said that the fluid leaves, but now I'm saying that it doesn't. So picture a tumor. You have like punch big holes in those vessels in the tumor. The fluid is going to push out. It doesn't get taken away, and that's going to make the difference in pressure between in the tumor and in the vessels. The difference is not going to be as high. Does that make sense? Everybody? Okay. So because of that pressure equalizes more, it's harder to get things out of the blood supply and into the tissues, right? There's not that pressure difference forcing it out. And so you get less diffusion of small molecules, and almost all of the chemotherapy drugs are small molecules.
So they're not able to...
Yeah, so it's harder to get them out. Now, the fact that the vessels are leaky helps, but the pressure difference sort of fights against that. Right? So on one hand, they're leaky, so you would think, oh, well, that would let things out. But on the other hand, you have this resistance caused by the lack of pressure difference. Okay? And so then what we're going to talk about is the angiogenesis inhibitors now. And so for those of you who were able to watch the video, the Cancer Warrior, I guess, Yalda, you probably didn't see it because you probably didn't get the thing. So I'll send you the link is on here now, actually, so you'll have it when I post this video. But it's about a 1 hour Nova. Nova is a science series on the public broadcasting. And so it's a 1-hour Nova called The Cancer Warrior. And it's about this man, Dr. Judah Folkman. And so, again, I was talking with the ITESM crew before you joined us that Dr. Folkman passed away in 2008. And he was still actively researching angiogenesis. It's very rare that you can look at an entire discipline of science and say that one person started it, right? You look back, who started physics? No one, right? There's no one person, right? People were observing it, but it's pretty clear that when it comes to angiogenesis and cancer, this man is the father, the grandfather. He is everything for the beginning of this discipline, which is angiogenesis. So in 1971, which was when Folkman first published his theory on the role of blood vessel development in cancer development and spread, there were 3 articles in PubMed, right? If you did a search, there were 3 of them. 2 were from his lab, okay? And probably the 3rd one was someone saying that his 2 were wrong or something like that, okay? So in 81, in 10 years later, there were 34. Last year, there were over close to 7,000 of them. And if you do a search of angiogenesis with cancer, there were 37,000. Okay, so this is a huge field now. Okay, the anti-angiogenesis drugs, the goals, right? You can prevent the remodeling of all that extracellular space, that environment that we talked about last time. If you can prevent that remodeling, then there is essentially a wall of fibrous tissue that the blood vessels cannot penetrate. Right? There's always connective tissue and extracellular matrix around all the tissues and cells. So if you can prevent the remodeling, then you can stop that. If you can block the endothelial cells from dividing and forming tubes, if you can block the factors that stimulate angiogenesis, what one would you target? What's the biggie?
The VEGF.
Yeah, VEGF, right? So VEGF, right? That was and is one of the major targets, right? Is to antagonize these. You can try to block the action of the integrins on the endothelial cells to block cell-cell connections. And ultimately our goal, right? What people are now looking at is not in blocking angiogenesis altogether, because I'll show you why, but maybe what we want to do is to restore a normal structure, right? It's kind of counterintuitive to think that if you can fix the defects in the blood vessels in a tumor, that you can actually make the cancer go away, right? It seems like that you'd be helping them, right? But it turns out that when you try to just destroy them, it doesn't work. And so now people are looking at what's called normalization. They're trying to maybe make those tumors act more normally. And that would help delivery of anti-cancer drugs and maybe even help with metastasis. Okay? So at the time of his death, right? So in 71, there was no such thing as angiogenesis. By 2008, over 1 million people had been treated with angiogenesis blocking drugs. And there's still a tremendous amount of research going on to them. Okay? It is something that is common to all cancers, right? We say cancer is so variable, cancer is so different, all cancers are different. That's why it's so hard to treat. But all solid cancers, and we now recognize that even a subset of the lymphomas and myelomas, all depend on angiogenesis. So it is a very exciting. If you're a drug developer, this is something that would be very, very appealing, right? You could treat all cancers potentially with one drug. Everybody see that?
Yeah.
Okay. So there are over 3,000 clinical trials listed in clinicaltrials.gov. When I did a search with cancer, there's about 400 phase 3 trials. And we'll talk about clinical trials. Phase 3 means that they have shown that this drug it works in a small group and it's not too toxic. And now they're looking at a bigger group to look for more rare side effects. Right? If we only look at 10 people, we may not know that it kills 1 in 11 by causing their head to explode. Right? So you have to look in a bigger group. Right? So in a phase 3 trial, you've shown that this thing has some effect. It's efficacious at some level, but you want to try it on more people to see how well it works, and you want to make sure that it's not going to make people sick. Okay? So that's very exciting. Okay? So the types of cancer that are actively being studied in phase 3 trials of angiogenesis inhibitors: breast, colorectal, stomach, liver, lung, multiple myeloma, lung cancer, ovarian cancer, oral cancer, prostate cancer, renal cancer. Okay? So there are a wide range of cancer types, right, that are being examined now for their ability to be treated by essentially starvation, right, is what we're talking about. Okay? And one of the key differences in angiogenesis treatment versus treatments like chemotherapy is that we're typically not directly attacking the cancer cell. We're altering the activity of cells that are in the stroma. We're altering endothelial cells, right? These are not the cancer cells. So because of that, they're not necessarily genetically unstable. Does that make sense to everybody?
Yes.
Right? The cancer cells are the ones that are unstable. So this looked like essentially a perfect sort of treatment, because we're not going after these cancer cells that are so slippery and changing. We're going after the cells that are making blood vessels. Okay? In 2004, the first of the angiogenesis inhibitors was approved. It's called Avastin. And Avastin is an antibody that is directed against the growth factor VEGF. Right? So this is an interceptor. The idea is that the tumors probably have lots of VEGF in them. They're inducing nearby cells to make VEGF to help them. And this antibody goes in there and it binds to the VEGF before VEGF can bind to its receptor. Does that make sense? Also in 2004, another antibody was approved called Erbitox. Erbitox goes against the receptor. So this antibody is directed against the epidermal growth factor receptor. Okay. Endostatin. Anybody ever heard of endostatin before?
Yes.
When did you hear about that?
in the Cancer Warrior.
Okay. Yeah. So endostatin came out in 2005. It was not. Sorry, y'all. That's completely, of course, your fault for not reading my mind and seeing that. Endostatin is approved for use in China. It is not used in the U.S. It's a very difficult drug to work with. It's highly hydrophobic. It sticks to tubes. The experiments are very hard to do. Endostatin isn't used here, but it is used abroad. Yeah.
Does China have less strict regulations?
I don't think they have any regulations.
So that's why they use more things.
Yeah, I mean, yeah, they do have. Yeah, I'm kidding. Of course, they do have regulations. They just don't seem
Oh
to.
yeah.
They just don't seem to pay attention to them. Yeah, so they do have less rigorous testing than the FDA, and I think they're much more. They're just more lax about accepting, negative results and things like that.
And what do you think about that? Do you think it's more beneficial, or they should be
I
looking at?
think I think they are. I don't think they really do what they should do.
Okay.
Yeah. I mean, the FDA definitely, people bitch about the FDA here all the time because if you're really sick, like Ebola, right? Like you see with Ebola, people say, "Give me the treatment." So it's not approved. Say, I don't give a shit. Give it to me, right? I have Ebola, right? So they don't. People are willing to try stuff because they're very sick, right? They figure that the alternative is not so good, but. The FDA is very, very strict about that. In Australia and the EU, they seem to be a little more lax, but also more like the U.S. system. So yeah, I don't know. Yeah, I mean, China doesn't seem to regulate anything the way I would want them to. So okay, in in 2006. A drug called Revlimid was approved, and so I put this up here because it is derived from thalidomide, and so for those of us who did their homework, we learned all about thalidomide and why thalidomide is now used to treat cancer. And so thalidomide, for those of us who didn't do it, I won't mention any names, Yalda, but it was not her fault. She didn't know.
Yeah.
But anyway, thalidomide was known to be something when they were studying this, and they show it in the documentary. It's known to cause terrible birth defects, right? But it also is now used to treat cancer. And so there's another one called Thalomid, which is another brand name of thalidomide, which is used to treat cancer. In your book, in the movie, they showed a guy named Dwayne Gay. Yes, the TV anchor.
Yes.
You guys remember that?
Yes.
Right, and he was taking the thalidomide, right?
Yes.
And so this thalidomide does have side effects, but it does block angiogenesis. Sutent is another. This one is a kinase inhibitor. It blocks the activity of the receptor. This is a list here of approved angiogenesis drugs, and so there are more than I listed. Many of them target VEGF. That some of them target the receptors for these things. It's important when you look here. I'll highlight. I don't know if I. No, I don't. I'll highlight one of these. You see, it doesn't matter. It doesn't really matter which one. But let's look here at sorafenib. You see sorafenib, where the cursor is.
Yes.
Okay, and I want you to see something about this. It's a tyrosine kinase inhibitor. It inhibits these receptors for the growth factors and others. Ooh, what happened? I don't know what happened. But where is it? Here's sorafenib. And what you'll see about this is it inhibits not just one receptor, but 1, 2, 3 receptor tyrosine kinases, as well as KIT, which is a kinase, and RAF, which is downstream from RAS. So what we're learning is that some of the more effective drugs actually, so let me back up. If you're a pharmaceutical company, let's say I'm a pharmaceutical drug developer, the mantra in pharmaceuticals has always been to develop the most specific drug you can. Why? Because if you develop a drug that interacts with 2 or 3 or 4 enzymes or pathways, you're going to definitely see more side effects. Does that make sense?
Yes.
Okay, so the pharmaceutical industry, their whole paradigm, right, their whole way of generating drugs has always been to find the most exquisitely specific inhibitor that they could come up with, right? And you see why that is. Unfortunately, in cancer, what we're learning is that if you block one particular angiogenesis pathway, by the time you get late in disease, when someone has a big tumor and has metastatic disease, they often have turned on multiple different pathways. And so even though these drugs may have more side effects, I'll show you later, what we're learning is that it's probably better to have drugs that actually interfere with more than one pathway. Because if you don't block more than one, these advanced tumors just will go around the one that you block and use a different one. Does that make sense?
Yes.
Okay. Okay. So I wanted to point out briefly, right, that this idea of angiogenesis is not just effective in cancer. There's multiple different things that have defects in cancer. One of them, which is macular, it's called a macular degeneration, a particular type of blindness.